Thank you very much for that introduction. As you see, I've changed the title a little bit, but we're trying to be within the same scope. And it's great to be a big room of friends and so many faces I recognize and friends I haven't met yet. So I think this it's also been some really good talks already. So we'll try to see where we fit into that scope and see if this works. So we do a lot of antibodies. When I say antibodies, they're, the family of antibodies now is just growing almost by the day. So they're all these different formats and different structures, different functions. So just from a semantic standpoint, when I say antibody, I really have not the same. This is not the textbook definition of antibody, but all these weirdo Frankenstein molecules that are floating around now. So we end up doing, I'd say, the majority of work we do is really in bispecifics and a lot of these Frankenstein molecules. But to set the stage a little bit, it's been really interesting to see over the last, I guess, decade now, how this whole industry has changed fundamentally in the world of pharma. So yellow here means small molecules, and pink are biologicals, or biologists. So we went from in 2010, all the top 10 drugs were all small molecules. And the most recent data I have is for 24, where 7 out of the top 10 are biologics. So it's really fundamentally changed. Of course, biological molecules are very different when it comes to production, when it comes to QC, when it comes to navigating biology to make sure that you generate the right molecule and glycosylation patterns and charge variants, and we'll get into that. And I think the other side of that is if you look at sort of the the growth of this market is really very, very different. So CAGR, so annual growth rate for small molecules is expected to be around 2-3%, whereas for biologics it's in the order of 10%. So really, this is the market. This is where therapeutics are moving, and this is clearly where there's a lot of opportunities to make these drugs. Having said all that, biology is hard. It's really complicated, and the numbers I've seen, and of course, it's going to depend on who you ask and when you ask them, but typically 90% of all the drugs that come to phase 1 still fail, right? So this is, of course, the absolute majority failed long before phase 1. So we're sitting here on a path to make molecules where basically everything fails, right? So why do they fail? Well, a lack of efficacy is a big part of that. Toxicity, immunogenicity is also a big part of that. Developability is a significant part. And various commercial issues, that's the part I'm not going to address at all, because that's a separate headache altogether, and it's probably a completely different conference. So how can we fix that, right? So I think that, and the other side is we need to generate these, and I think a couple of people already mentioned that discussion in the sense that it's really slow and it's very expensive to generate these drugs. So how can we make the process much faster, much more efficient, and much more predictable, right? And the other point of this is really coming to an antibody conference. I think that I've never seen a drug fail because it doesn't bind. So most of the talks are typically around binding affinity and KDs, but that's I think that's necessary, but certainly not sufficient to make a good drug. So, and here I'm guilty as charged, but I think we as scientists, we got to think about why are we here? What's the point of this conference? What's the point of coming to work every day? It's not to, as a nerd, I tend to dig into, and those who know me, I go on for hours on codon optimization and single peptides and all these different things. But really what we need to do is just take a step back and say, what are the key criteria that is limiting the growth of this industry, and how can we make our little dent in these main issues? So what I'll talk about today is really how we address process, so make it faster and more predictable. I'll also address lack of efficacy and how we engineer that from an ATUM perspective in one nice little case study, and also developability as a nice little case study. So wrap those. So it'll be sort of 3 stories put into one. So for those who don't know ATUM, we've been around for 20 plus years. We make a lot of millions of genes, 10 to the 4 of proteins, 10 to the 2 of cell lines, and been doing that for a long time. We are 150-ish people over in California. One of the big, let's see how this works now. Anyway, so one of the key differentiator for us, going back to some of the discussions earlier this morning, the company was really founded around building our own LIMS, our own laboratory information management system. So we started with the IT, and then we built a company around that, right? So one of my co-founders, Sridhar, is a database guy. So day one, we were 4 of us in a little office. He started writing out on a blackboard, sorting out what the different columns should be in the underlying database. So we have now, in the order of just looking at antibodies, for instance, we probably have 200,000 antibodies where we have sequence and titers for, right? So, and captured within this underlying database. I think that is one really cool differentiator on how we approach these problems. So that becomes, taking all that data now, we can do a lot of machine learning and AI and trying to figure out what works and what doesn't work. So here are a couple of tools I'll talk about. So I don't dare to press these for, okay. So left to right. So one new thing I'll talk about is what we call RNA 2.0. This is sort of to our old DNA 2.0 heritage, but started making RNA and how that helps out. I'm not going to talk about gene GPS, but with lightningHEK is a nice new system. We do linear DNA that used for fast, efficient antibody production. We're still getting all the PTMs and all the biology happening on those sequences, but it's really fast. It gets you to a number of antibodies quickly. We built a separate one called discoCHO. I'll go quickly through that one, where we have a transient system for making antibodies that look very much like what you end up getting in the final CHO-K1. And of course, we have the leap-in transposase miCHO system that provides you the sequences that you go straight to the CMO. And going back to this is really trying to build this whole pipeline with the end, beginning with the end, right? So how can you make all the molecules at the end going to be made by CHO-K1 in sort of a large kilogram scale? How can we make sure that all the previous parts in that integrated chain fits that scale and that system and those post-translation modifications, those charge variants, all these different sequences? And how can we make that as integrated as possible and as information rich as possible? So let's start with RNA. So for those who know us, we've done a lot of work on leap-in and transposase for making cell lines. And when you make the transposase, you typically co-transfect a plasmid that contains typically the antibody or antibody-like molecule with a piece of RNA that encodes the transposase, right? So historically, we used to purchase this RNA from a couple of different vendors. Problem, those who didn't get RNA from outside vendors, you know it's slow, it's tedious, and honestly, the quality is not that good. So we decided to bring that in-house. Made me very happy. I spent more years than I like to admit doing it in vitro transcription back in grad school and postdoc. So it felt like coming home. And it allows us to have the full process in-house. So here's just an example where we do, oh, it does work. Here's an example where we took the vendor and we tested in fluorescent protein in HEK and in CHO. And this is the exact same sequence, 2 different constructs we made. And this is the external vendor that we compared to, that we used to do for all the leap-in transposase RNA. Now we moved all that stuff in-house because you just tend to get much better, higher quality sequences, much more robust signal. And it allows us, because we have the entire process, of course we can process the plasmid DNA, we can do all the QC in-house, so we can really have everything battle tested and QC'd. So one of the things we built really is poly A. Turns out coli does not approve having 100 A's in a row. Coli tend to toss those things out. So we built our own little coli system, also with some really nice assays to be able to quantitate this at a high degree. And also we do enzymatic capping instead of all these synthetic caps that other people use. And so works really well. We talked to some of our friends. So I don't know if the mAb people here in the room, but this is really cool. So they've been using RNA for immunization. Historically, as many people talk about here, you get protein and you kind of purify it out to be able to get a good antigen for your antibodies. And there are all these issues. Sometimes that's easy, sometimes it's hard. So this is for a transmembrane protein that mAb trying to get antibody for. So here you compare what their previous RNA provider did, and now with the RNA that we did, it's a much faster response, and of course much higher response as well than compared to vendor X. So this, I think that from a production of antibodies, it kind of hops and skips over the whole process of making the protein. You can just go straight making the RNA. That's very straightforward. You never have to worry about all the different issues that comes with purifying the protein. Just inject RNA straight into your animal. Boom, you get beautiful, beautiful responses. So that's for the RNA 2.0 part. And so next what we do is build a what we call a in silico AI down select of all, where you inject some antibody in here, or you inject some antigen into the mouse or your system of choice. You get a million sequences coming out of that by next-gen sequencing. We can use some AI. I'll talk briefly about that to down select all these millions of sequences to something you can test. And then we typically go straight into lightningHEK, which is this linear DNA that gives you 10 days from virtual sequence to actual protein, and then if we down select off of those and go into discoCHO, which is discoveryCHO engine, to make sure we get the same charge variant, same glycan profiles and then move that into miCHO. So that's really comes into the discussion of how do you make sure you have an integrated process where you drive that as efficient as possible, and making sure you always really start with the beginning with the end in mind, right? So how can we get to kilogram levels as fast as possible and as efficient as possible and capture all that information? So in the down select, there was some really cool talks this morning in this place here in this room. We have, as I said, lots of sequences over the last 20 years. So we built predictions. So this is an example of a titer prediction machine learning. This is just looking at antibodies and I think this is based around 20,000 antibodies. We get a cross-val here that is around 0.6, 0.67, 0.65. So decent, good correlation. So we can use this now to take all this in silico sequences we generate from next-gen sequencing, from the antibody, from the animal, and then just weed out everything that is supposedly a poor expressor. And we have also working on titer aggregation and other prediction models that we combining now to be able to weed through this as quickly as possible, right? And at the end of the day, underlying this is 200 to 250,000 wet lab antibodies that we generated and we synthesized and we built over the years. So once you've gone through that one, now we can put those sequences, so go from maybe from a million coming out of the mouse, maybe we'll take 5,000 or so and move them into lightningHEK. So lightningHEK is these linear expression cassettes. I think James talked about them earlier. He's in here somewhere, or he used to be. So that gives us 10 business days from a virtual sequence to a purified protein, typically 200 micrograms and up. And that allows you to go directly to developability testing on a small scale. And the next step is what we call the discoCHO, this discoveryCHO. So I usually introduce this as what Agneta and Annefrid used to say back in the ABBA days, give me a mAb after midnight. So I sang that at some conference. It was, I think my Alain still haven't forgiven me for that. So I won't do it again, ever. But discoCHO is cool because it provides the exact same quality attributes as you get from a CHO K1, right? So typically when you get, most people do their transient in HEK or in CHO-S. But those are not predictable on all the properties like the titers, the glycosylation pathways, the charge variants that you see from getting a CHO K1 as a transient. So this is exactly the same CHO underlying strain that we use for the stables as well. So it's as a stable, we call it miCHO, and for transient, it's a discoCHO. And this gives you from virtual sequence to protein in 4 weeks and allows for all sorts of complex biologics. And fundamentally, it's the same underlying strain, right? So it gives, and then we built this to work directly with off-the-shelf media, make it fast, make it efficient, and be able to scale that up. So here's just, I think this is I weeded out a lot of slides because I've only got 20 minutes or whatever it is. But here's one that I think is very, very telling. This is the charge variants where we look at transient and from a CHO-S, from a non-K1 CHO, compared to the miCHO, which is what you're going to get. This is a CHO K1, which is exactly what you're going to get when you do typical commercial scale. And this is the disco. So what you see here is the charge variants from an antibody, from a fusion molecule 1, fusion molecule 2. And you see almost identical patterns on the charge variants between the disco and the miCHO, and whereas the CHO non-K has a very different patterns in all these different instances, right? And you much rather want to know all the charge variant and all your distribution of diversity at this stage instead of when you're down starting to do stables and costs really skyrocket. And the last step on the integrated pipeline, of course, is the leap-in, the transient, the transposase-driven process we built for making stable cell lines. And it's now, of course, there's been quite a few companies over the last few years who are jumping into the transposase business. I think we're still, I'm biased, of course, but I think we're ahead and we do a lot cooler science and we're better looking too. So, but this is really leap-in allows you to really, this is back in the early computer days, what you see is what you get, right? So this is where Mac came in and you can actually design on the screen what you get. So this is the same thing with biology now. What you design on your monitor is exactly what's going to be integrated in the chromosome, right? So when you do all types of random integration, you get all these truncations and concatenators and all this garbage, right? So it's weeding through that is a mess. Transposons, you get this nice clean cassettes, pop in, pop in, pop in. You get perfect distribution, and you get most of your sequences going to have a really high titer, and you're going to have all consistently with already in the pool. You'll have consistent material. So we have a lot of our customers, they go straight from pool to tox studies because of the consistency. And during COVID, of course, we had a couple of pools go straight into man, which is cool. And so the stable cell lines generated using miCHO and leap-in, very fast, you can go to pools very quickly, and the pools are very consistent with what you get at the final clone stage. So it's a very efficient way to screen large number of constructs, typically high yield. The genetic stability is amazing. Transposons basically evolved over 4 billion years to be stable. That's their one selected advantage. So incredibly stable, homogeneous population, and you can do all the engineering you want. So we've done this now for quite a bit. We have a lot of molecules coming to market now in 4 different territories. So US, of course, Europe, China, and Australia have all approved, and the regulatory agencies seen and approved the leap-in platform. I think we have 45. Is that still valid, Alan? 45? Was it 40? Okay. 45 IND filings approved, a lot of licensees, a lot of top pharma, a lot of projects delivered. Right? So it's well established, well accepted by regulatory. We now have 5 different leap-in that are orthogonal to each other. So you can go in multiplexing them. You can go in with one, knock in the gene of interest, go in with a second one, knock out like glycosylation patterns or whatever you want. You can start really multiplexing that. That's a whole seminar all by itself. And Mario is in there somewhere, happy to talk to you about that for a long time. maybe a little bit longer than you want. Okay, so that's the integration. I'm running through a couple of slides here. Hopefully, I haven't seen the minute number come up yet. But here's a cool study. This is something we did with Lundbeck, and we talked about this one in the past. This is one of the things when you engineer for developability is that it tends to be this whack-a-mole, right? So you fix something binding, and now the aggregation goes to hell, and then you fix aggregation, and then immunogenicity goes to hell, or whatever it is. Right? So you end up doing this endless whack-a-mole. So we did this project with Lundbeck. They had a project where the binding to their target was negatively correlated with PK values, so in this case, as measured by a specific polar reactivity assay they had. So they want to engineer this enzyme, this antibody for at least these 6 different properties. And of course, this is in the public domain. You can find this paper if anyone's interested. So I just like this slide because it kind of illustrates the point of whack-a-mole. So here, the starting point is the green X, and the goal is the box, right? So we want to, this is the starting point. We want to move it into the box. So here we want to improve binding by a couple of orders of magnitude. Well, basically, there are more things here as well, but I'm just taking the top 4. So when we do this, we do it using AI and ML and ancestral reconstruction to identify what variables to change in. And then we use DOE, design of experiment, to systematically vary all those variables so we can maximize the information content. We're basically putting, in sequence function space, we're basically putting beacons that have the most information-rich nodes in that space. And then we use gene synthesis to make those exact molecules and then test them and then use that information to map out the fitness function correlation. So in this example now we're doing just small set. Each set is typically 96. So you see from round 1 we get a distribution in KD, we get a distribution in titer, and so on. And in polar activity we didn't really touch that. Okay, so now we can engineer for binding because we have a distribution. That means you can build a model. That means you can start navigating. And we get a little bit better. And then we do round 3 to really make the jump. And now we get a couple of binders that are within the goal. But at the same time, what happens, of course, is because we have this negative correlation between polar activity and KD, all the polar activity goes to hell, right? So that's crashes and burns. So this goes back to the whack-a-mole. And titer and TM, same thing. You see how you get the distribution. But because we have this nice distribution from really good polar activity to horrible, we can actually use that as a machine learning model to build predictive models on that and identify sequences that are that fulfill all these different fitness functions. In this case, actually, we're looking at 6 different fitness functions. So in the end, we get 4 molecules that are all within the box in these 6 different fitness functions that we work on. So that allows us then to really engineer for multiple developability properties simultaneously in order to maximize the function of the sequence. And point being, trying to do it as fast as possible, trying to do it with as few molecules as possible to maximize the information, sequence function information content for building these data sets. This is actually a Lundbeck slide. I don't know if there are Lundbeck people in here. I saw some of them earlier. But the total space we're looking at here is, because of the systematic variance, we can explore space that is 10 to the 27, which of course is just ridiculously large, right? So this is, I don't know, stars in the universe or something stupid like that. But because we're doing it with AI and ML data sets using a design experiment to search that space efficiently, and anyone who's more interested in that, come find me at the booth later. We explore a 10 to the 27 space, but only 400 variants over 4 iterations. And optimizing then in 6 different dimensions, right? And fulfilling all the criteria that we needed. So here's another cool example. This one is actually first time I showed this in public. This is with partner X. They came to us and we engineered an antibody for binding. So this is for viral neutralization. So this antibody binds to both of these 2 viruses, virus 1 and creatively named virus number 2. Those are related but different. And one of the first things we noticed is that it didn't matter. There was really a poor correlation between viral inhibition and binding. This KD, right, there's no correlation here between in either of these cases for binding and neutralizing the antibody, right? So binding is necessary but not sufficient in order to neutralize a virus. And viral inhibition the assay is miserable. It's horrible. It's error bars are this big. It takes a lot of experience to do these things, and ugly assays. Whereas, of course, binding is easy. You can just shoot that in a fax sorter. You're done. So we had to pivot here and start optimizing for viral inhibition, given here as IC50, for both of these antibodies, for both of these viruses. Sorry. So what you see here is in the round one, the first set of 96, we get a certain distribution for primarily for virus number 1 binding or neutralizing. That's my Swedish accent popping through. My apologies. And in virus 2, we also see it, but less of a distribution, so less of a model, right? And then we iterate that through in round 2 and round 3, and we end up getting molecules. The final molecule is improved for viral inhibition, not binding, right? Binding is binding. That's easy. But the real commercial endpoint assay is here, how can you neutralize the virus? So we improved that around 500-fold for virus number 1 and 200-fold for virus number 2. So now you can use the same antibody to track those two related. And again, the total space we're exploring is 10 to the 16. So very, very large, right? But we're only testing, in this case, around 300 antibodies over 3 rounds, right? So on top, I'm just looking at viral inhibition now, but also we optimize for titer, for aggregation, and for other features as well. So if I had a watch, I'll see that my time is running out. So really what we've focused on here for this talk is we're trying to make our little dent in how to make this process much more efficient, right? So we're building this system now with in silico down select, the lightningHEK, the discoCHO, and of course the miCHO leap in as one integrated pipeline that allows you to navigate the development of antibodies as quickly and as efficiently as possible, with as minimize the risk of because you're looking at all the right properties as early as possible. And with the GPS engineering platform, I showed you one example of how we really look at the efficacy of the molecule, not just binding, but really the efficacy of the molecule. We have lots of those examples, but typically those are cell-based assays that are much, much uglier than just shooting things into a fax order. And of course, the parallel developability optimization. And as an added bonus, just because I think it's pretty cool, I think that the RNA gives us a lot more, and I think goes back to the discussion around membrane proteins earlier. A lot of these proteins are horrible to work with, and you know it better than most. But if you do that through RNA is easy to make, very consistent. You can just generate antibodies, for instance, very rapidly using that platform. So with that, I thank you very much for your attention, and I'd be happy to take any questions.
We have time for one quick question, and I see it up here.
And you can always ask questions down at the booth, which is number
332. Okay, so booth is 332 for further questions.
Yep.
Absolutely fantastic talk. So you've illustrated beautifully that you can go from 1 million sequences to, 10 to the power 4 protein and cells. And then you made this point about just 45, was it 45 in the clinic? Sort of my question, and I don't know if you can disclose this, do those 45 molecules fall within certain companies, and are they doing something better than others, or is it spread all over the map?
No, so your question, if I understand it correctly, let me talk in front of a microphone, which probably make more sense. So if I understand you correctly, this is the IND filings coming out of the LEAP-in, right?
Right.
Yeah, so LEAP-in is a nice platform for making stable cell lines very rapidly at high efficiency. Those molecules are across the spectrum, everything under the sun that is antibody related. And we've seen everything from bispecific, regular vanilla antibodies, IG1s, IG4s, bispecifics, trispecifics. There's probably some talks later on at this conference. I know at least 2 talks where they They may or may not disclose that we made their constructs for IND filing. So among those 45, there are probably 20 or so companies. So it's fairly distributed. It works really well. It's become very rapidly the sort of the default solution for a lot of these antibody production for cell lines.
So I guess the point being, you can definitely, get some information, because if you're removing that variability at that last stage, right? I mean, clearly you can start connecting the dots, which I think is just fantastic, right direction.
Absolutely. Yeah, that's one of the key points with building a streamline. I mean, it goes back to some great talks earlier today around the complexity of building these large confederate data sets or protein expression. I think that this big struggle we've seen, I mean, we see differences from Kuhner Shaker 1 versus Kuhner Shaker 4, right? So can you imagine then if you do this at ATUM in California versus, BioInvent in Sweden or whatever, right? There are all these, everyone's got their own humidities and people pipetting differently and all these different things. So there are so many different variables that are really, really hard to control for. So what we like to do is just have everything as standardized as possible. May not be the best process for every single molecule, but what we're trying to aim for is the best possible compromise that allows for the most majority of molecules to be processed as efficiently as possible towards the final endpoint, if that answers your question.
Okay, let's go ahead and give Claes a thank you.